Right. Good morning, everyone. Welcome to this month's monthly seminar. Just to remind everyone that we are recording this. It's my great pleasure to introduce Alan Radford from the University of Liverpool. And I'm very jealous of Alan because he's actually discovered a new coronavirus as well, which I think, well, a dog coronavirus, it is a new thing, a new strain, new variant, Alan. And also he's been pioneering the surveillance of diseases and the health of small animals, which he's going to tell us about today. So over to you, Alan. Thanks very much for agreeing to talk this month.
Right, let's get back to the beginning. So I'm going to tell you a story. Who was that? Tommy Cooper. I guess some people on the call, if I'm right in it being Tommy Cooper, then there'd be plenty of people on this call who have no idea who I'm talking about. Julian, is it Tommy Cooper? You're my age.
I can't remember, but you need to wear whatever hat he used to wear, but don't Fez, but don't die on stage like he did, Alan. Just
okay. I'll
make
try
it
not
through
to.
for another 25 minutes.
That would be awkward. It must have happened on Zoom at some point. And so I'm going to take you through we don't use the word pandemic, obviously, we use the word outbreak, an outbreak that occurred in domestic dogs in the UK in 2020, and share with you some of the work we did on a very tiny scale compared to what was going on almost in parallel for COVID. But we were doing it on an absolute shoestring. And I think for me, it shows some of the powers of the technologies we have now available to us, which are increasingly accessible to small teams of people. You don't have to be massively funded to do to use some of this exciting technology to do some quite nice science. But it's going to crash together some areas of research that many of you will be used to if you're a coronavirologist in terms of sequencing and phylogeny with health record surveillance, which is obviously quite different. What you'll see in this story is how we crash those two things together. We're going to talk a little bit about the virus the variant we've discovered, and we're also going to finish by talking about some of our attempts to use AI to spot these outbreaks earlier. So AI of health records and whole genome sequencing of samples to better understand coronavirus epidemiology in dogs. My name's there. Happy to get emails afterwards, but very much representing a team, and I'll introduce you to some of the key people as we go through. Although this work wasn't specifically funded, the group that I'm lucky enough to be part of has had most consistent funding from the Dogs Trust over this period, and very grateful to them for that support. So, canine enteric coronavirus, it's an alpha coronavirus. It's very closely related to feline coronavirus, perhaps the better known companion animal coronavirus, which is often called feline infectious peritonitis virus. I think you can see my mouse wobbling, but maybe I'll go for the laser if I can find it. There we go. There's feline infectious peritonitis virus, the better known small animal coronavirus. And just above it, you'll see canine enteric coronavirus and swine, one of the swine and pig coronaviruses as well. Very, very close related, and they've had this long, complex history of sharing bits of their genome. And I suspect in the fullness of time, we might stop calling them pig, dog, cat, and call them some kind of animal coronavirus, because it's clear they have a very mixed evolutionary history. In terms of disease in dogs, I lectured to our vet students about viruses that cause diarrhea in companion animals, and previously I would have had probably 1 or 2 lines in a whole lecture on canine enteric coronavirus. We've known about them for a long time. They arguably we think generally were causing nothing, but perhaps very mild diarrhea. There were sporadic reports, publications of dogs, usually very young dogs, dying of diarrhea that were testing positive for canine enteric coronavirus. But these were individual animals, young animals, and usually co-infected with something like parvovirus. So it was was a little bit tricky to know exactly what was going on. And so we generally considered canine enteric coronavirus as an insignificant or at best cause of mild diarrhea. There was a vaccine, or there is a vaccine that was licensed in the UK, but because of probably my lectures and other people like me saying it's not really a big deal, the vaccine was very poorly taken up, and was discontinued in the UK a few years ago. Okay, so that's the history. Now, the date here is very important. So we've got time, but let's just deal with the date. 6th of February 2020. There were local reports of, and this started, the very first report was from a vet in Liverpool, of vets saying, I'm seeing a lot of dogs with vomiting. And the vomiting is more severe than we would typically expect. So if you have a dog, you will know your dog will vomit. They're obviously not feeling particularly sick because they'll vomit and go and have their dinner. They'll vomit and eat the vomit. I mean, that's what dogs do. They vomit once quite frequently, but this was 10, 15 times vomiting. These dogs were very, very depressed. The veterinary term depressed, not the human term depressed. And they would take several days to a week to get back to normal. Some needed hospitalization. There weren't any credible reports that the dogs were dying of this thing, whatever it was. And obviously, February 2020, 6th of February, let's say that's about here. And the big curve that you can see there is not what happened to dogs, but that was human coronavirus. So we were at a very stressful time on the planet. And there were questions about, could this be to do with this funny virus that people are talking about in Italy? Because we were hearing about this in the news in Italy, for example. And so in companion animals, there is no national surveillance. And so it's really, really tricky to know whether these anecdotes are real. We've had them for years. Anecdotes have increased this, increased that. And it's impossible to know. Somebody needs admitting, if someone could admit them. It's impossible to know really whether those historically have been real events. But we've now been working at Liverpool as part of a team called SAVSNet, the Small Animal Veterinary Surveillance Network, to put in place national surveillance of companion animal disease using health records. So if you have a dog, or a cat or a gerbil or a hamster, when you go to the vet will type stuff into the computer. That's a health record, and we collect those across the UK. We've been doing it since about 2014 in a big way. We have about 12 million records, and we can look in them for patterns of disease. And one of the reasons we set it up was to look out for outbreaks. So we thought, let's have a look. Vets are talking about this. Let's have a look. We have lots of signals that we can look at, but this is the main one that I'm going to concentrate on today. At the end of each of those 12 million consultations, a vet is asked, what's the main reason the animal's here? It's a single question. It has to be really quick, simple, because these are busy vets. And one of the choices is gastroenteric disease. So this plot here is showing the pattern of vets saying the main reason this animal was here was gastroenteric disease. And you can see 2018, 2019, a gentle seasonality, perhaps slightly worse in the winter, gentle seasonality. But at the beginning of 2020, something very different has happened. So this is gastroenteric disease dogs. We wondered if it was something to do with our health records. Were they being collected differently? This is respiratory disease in dogs, very flat. This is gastroenteric disease in cats, very flat. This is meropitant, which is a drug that vets use to treat vomiting, and the sales prescriptions of it. And again, you can see the same pattern, and it's really nice when you get these two can't be a triangle. What do you call it when there's only two of them? Two things saying the same thing. I'm going to show you a third in a minute, then I'll use the triangle word. So lots of meropitant in dogs, not in cats. We get the free text that the vet writes. Saw a dog, it was vomiting. So we can look in that for vet using the word vomiting. It's a bit more complicated than that, but we can do that. A noisier signal, but the same. And this was looking not just for vomiting, but for frequent vomiting or severe vomiting. The pattern was all over the UK. So it wasn't just a very local outbreak. So at that point, we thought, well, I wonder what's going on. SouthNet, as well as collecting data from vets, also collects data from labs. And so we collect probably about 60 or 70% of the diagnostic tests that are done on dogs and cats and gerbils and hamsters in the UK. And they're tested for all sorts of things, including pathogens associated with vomiting and diarrhea. I don't know, can you see this bar, this muting? Can you see at the top of my screen, this meeting is being recorded? Because it's hiding. No, you can't. Okay, thanks.
So we can see your stuff, Alan.
Yeah, thanks. Thanks, Julian. I keep getting lots of pop-ups from Zoom, but I don't think they're showing on the screen. So this one is looking, the most likely virus to cause this was parvovirus, and this was parvovirus. The blue line at the bottom is the numbers testing positive. No, the percent testing positive, sorry. And that was very flat, but canine enteric coronavirus, which causes nothing, but was still being tested for, had a seasonality that fitted with what we were seeing. So this was January 2020. It wasn't any bigger than usual, but it was at least seasonal. So my colleague, Shirley Bonner, fantastic Shirley Bonner, decided to ignore what I'd said, which is very sensible usually. And she decided to test them by samples that we were now receiving from affected dogs by PCR for canine enteric coronavirus. And she was able to find that most of the samples we were receiving were, and this is M-gene Sanger sequencing, were a single, we now use the word variant, of canine enteric coronavirus. There were some others, you see down here, dog 15, dog 16, but the vast majority of animals were infected with a single variant. And that was already unusual because you don't tend to get outbreaks of a single variant of canine enteric coronavirus. We whole genome sequenced 6 of those to see whether the M gene, because it's only a small, I think the sequence of the M gene region is about 400 bases. So we sequenced the whole genome to see whether this phylogeny was representative of the whole genome, and it was. And here we can see these major variants are spread across the country. So the blue, the colours are the same as on this phylogeny. So this major variant in blue is spread across the country. Again, something that we weren't used to seeing. Okay, so that was 2020. What we were, I'm just going to jump out of PowerPoint for a second. So we then set up, and this is a website that you can go to if you're bored sometime. So this is live data. If I click on cats, you'll see it changes. This is looking at those 12 million records over time, and these are the different options that vets have at the end of the consultation. Why was the animal here? So I'll put it back on dog and gastroenteric, and you can see nothing much going on up to January 2020. There's this is lockdown. This little spike here is lockdown. You can see the number of consultations that we received massively reduced because of lockdown. But our outbreak preceded lockdown. What we wanted to know is what was going to happen afterwards. And you can see that in the 2 winters that followed, January, February, and January, February, we got the same, a smaller but a similar pattern, different to pre-outbreak, not as big as outbreak, but we got a similar peak. You'll see on the next slide, vets started to talk about this in Yorkshire, and we can zoom in on Yorkshire, and you can see that in January 2022 in Yorkshire, things seem to be very narrow peak, but quite bad for gastroenteric disease in dogs. So jump back to the talk, and you can see we're now in January 2022. Again, vets were talking about mystery dog vomiting virus spike in Yorkshire. So we were proactively at this time trying to collect samples during that outbreak. We did the same again. We got around about 50 samples from vets and around about 100 samples from diagnostic laboratories. And again, we sequenced them. The pattern was broadly similar. So there's a lot of data on here, but the take-home message is relatively straightforward. So this is M gene phylogeny. I'll put my laser back on, it's easy to see. This is the 2020 results here. So we had a single major variant, which is shown by this, and then a couple of minor variants. We call them minor variants because they were rare. And on the picture over here, you can see where those viruses were in orange. Go forward to 2022. Again, we had a major variant. So in this third winter peak, we still had a major variant, the dark purple, and it was spread all over the country, dark purple. But we also had much more variation in the population in this outbreak period, as shown by these, which we call minor variants, these light purple sequences, which you can see distributed. So many variants, but a single major variant. How was it related? Was this the same virus, the one here? Was it just hanging around and coming back? There, the M gene's not good enough, so we were whole genome sequencing, and what we were able to show was that the major variant from 2022, which is what's being compared here. Here we're looking at the whole genome sequence of that virus, and we were able to show that there was a big recombination event in the spike gene. The parent of that recombinant, or at least the closest sequence we could find to it, was a published sequence. It's actually quite old sequence now. You can see here A76, which is the closest over the recombination area. So we were thinking that we didn't have history in 2020, but we had history in 2022, and we think the 2022 spike was a recombinant. What was in the spike region, and the rest of the genome? Here we're using whole genome sequence to look at what was going on there, and this is the major variant from 2022. This phylogeny is masked for the recombinant regions, so we're just looking at the regions that aren't recombining, which we think is the best way of looking at the history of the major backbone of the virus. The major variant from 2022 was very closely related to the variants from 2020, which you can see here. That was done with a combination of CRISPR and amplicon tiling with great help from colleagues in CGR, and some interesting methodology there, but I probably don't have time to go into it now. But if someone wants to ask a question, very happy to. I.e., why CRISPR and amplicon tiling? There have been reports in between 20 and 2022 of human variants of canine enteric coronavirus. Previously, it's the dog virus. It's not zoonotic in any way, but there are rare but headline-making reports of human variants of canine enteric coronavirus, often coming from overseas. So we're obviously interested to see whether what we were finding in the UK could potentially be zoonotic. The zoonotic ones tended to have significant deletions in the spike, which we weren't seeing. So we were reasonably comfortable at this stage to say that they probably weren't zoonotic. And there was no evidence of zoonosis. What we've been doing since is, so we've sequenced in outbreak periods in the winters, and what we wanted to know really was what was going on in the summer preceding the outbreak. Our hypothesis really was that there would be masses of variation in the summer, and then a single variant perhaps has been picked to cause the outbreak in the winter. So we started to collect samples just after the 2022 outbreak, which you can see here. We don't call it an outbreak now, actually, it's just part of the seasonality. of the virus. We started collecting in the summer, I think, of 2022, and we collected for a full year to collect through the next outbreak. Well, the long and short of it, we didn't really get an outbreak in the following winter. We got a slight increase, but a slight seasonal increase, but not in the same way. And when we looked at the sequencing of these viruses, and these are the yellow ones collected, I think we aim to sequence about 10 a month over the full year, and there was no major variant at any point. So very different epidemiology of following 3 winter peaks. And we think that's probably, again, all speculation, but probably something to do with the dog population now being relatively immunologically competent to the previous major variants, and a new period of perhaps diversification beginning. And maybe in the future, a new variant will emerge that can cause that severe spike that we saw in 2020. We're parking that work at the moment, but Richard Abanovitz is starting to look at the immunology to see how these variants and the dog's immune response get on together, which is quite exciting. So a quick jump to something else for about 5 minutes, if that's okay with the organizer. Might pause.
Yeah, that'll just give you 2 minutes for questions, though, Alan.
Okay, so wait, not you, Julian. Something popped up on my screen. So what one of the things we're keen to do is to look for new ways of spotting these outbreaks that don't rely on vets clicking a button. So this is PJ Noble. If you want to know more about AI and text and how you can use AI to get meaning from text, then I would thoroughly recommend a chat with PJ. We've got 12 million records, 12 million texts. We can't read them. Can the computer get meaning out of them? Well, it can, using a thing called topic modeling. One of the topics, and we don't have to tell the computer anything to do this, that it finds, patterns of text that it finds absolutely mirrored the outbreak. And the topic is driven by these words which have diarrhea and vomiting in them. So we think this topic, had we had it, would have spotted the outbreak just as well as vets clicking this gastroenteric button. Working with some more sophisticated technology using large language models and a model that's been developed based on PubMed, but has been refined, hence PetBert, using small animal clinical records by, again, PJ and his PhD student, Sean. It's part of the BBSRC Doctoral Training College, and Sean's based at Durham. He was able to show that with PetBert, we actually would have spotted this outbreak, hindsight's a wonderful thing, 2 weeks before vets clicking the button. And that's a really exciting opportunity for the future, where we try and spot outbreaks in populations that don't have good surveillance. So to finish off, I won't read through the conclusions, but the last one, just be careful of fake news. We've always been very careful about how we talk about canine enteric coronavirus, especially during 2020 and the pandemic, because and I definitely never said this, Liverpool professor offers advice on mystery illness in dog confirmed as COVID, and we never said that, and the Echo took it down. So thank you very much. I'll stop at that point.
Thanks, Alan, and thank you for ending on fake news. That was a really fascinating tour de force of work. If anyone's got any questions, please put your hand up or put them in the chat, and I'm happy to put them into Alan. Alan, I'm going to start, if I may. You show variants, the same variant going across the country with dogs. What's the mechanism of transmission of that? Is that movement of people? Obviously, we're movement of dogs.
Yeah, I mean, I think I guess we would have to say that's likely what it was. Obviously, the first outbreak was very well. An actual outbreak in 2020 was very closely followed by lockdown. So we were we had no way of studying it, but I suspect that had a massive impact on the spread. And we wondered whether our outbreak disappeared because of lockdown for a human virus. In the 2 winters that followed, I don't think we were in periods of lockdown, certainly not in 2022. So there was movement. People do move with their dogs. They take them on holidays. And I guess that's what we'd have to speculate. There is also a wildlife angle to this because canine enteric coronavirus has been found in other carnivore species, funny enough, in Wuhan. I think a part of their enhanced surveillance of coronaviruses there, they picked up canine enteric coronavirus, Chekhov, we call it. Some people might remember Star Trek and the guy called Chekhov, or Seekov. Picked up variants in, I can't remember what species it was, but wildlife in Wuhan. So maybe wildlife has a component to play as well.
Okay, thanks, Alan. I'll move over to David. David, you got your hand up, and then over to Dan.
Thanks. It was a really interesting talk. It's amazing to see how everything kind of pulls together from all the data to the sort of surveillance and sequencing. Can I ask a naive question? With canine enteric coronavirus, do you expect that these kind of recombination events are happening all the time and it just happened in this case to be advantageous? Or is this something more unusual? I mean, if you think about SARS-CoV-2, for example, or human coronaviruses, they don't necessarily recombine all that often.
Yeah, I think it's quite a common part of the evolutionary history of that little group that I showed at the beginning of TGEV, the pig-1 feline coronavirus or FIP virus. Probably should call it feline coronavirus and canine enteric coronavirus. They have a long history. It's made the nomenclature actually really complicated, which I personally don't like. But yes, it's been there for a long time. There is also, you may have come across this, let's use the word variant again, of feline coronavirus in Cyprus, which seems to be very unusual in that it's causing outbreaks of FIP. You don't normally get outbreaks of FIP, you get individual cases, but this is an outbreak of FIP-like signs in cats. And that is, again, a recombinance between a feline enteric background and actually one of these canine enteric coronavirus is isolated from a severely ill dog that I mentioned at the beginning. So yeah, I think it's what they do.
But thanks for the question, David. Thank you, Alan, very much for that fascinating thing. And thank you also for pointing out about the feline coronavirus in Cyprus. We actually, as a network, had and I think you spoke at it, didn't you? A small group workshop in Cyprus around the biology and the genetics and the treatment of feline coronaviruses. And it goes to Dan's question in the chat about, is there an appetite or need for repurposing COVID drugs for these dogs? I would encourage anyone to look up about repurposing the drugs for cats, because basically it's a story of little old ladies meeting Walter White and Breaking Bad and smuggling millions of dollars of human drugs into the States to treat their precious cats. With that to do, thank you, Alan.